Okay, let's get started. So in this lecture, we'll be talking about transcription factors. A little different from the previous several lectures, which we heavily focus on algorithms, especially how to analyze the sequence data. So the transcription factors will be started by talking about the biological concept, and then several related important concepts and related computational methods for the analysis of transcription factors. So a brief outline of today's lecture. So I'll give you a very brief overview about transcription regulation and why this is important in biology, in general in biomedical sciences. And for transcription regulation, the key player in this whole process is transcription factor. And transcription factor is a type of protein that interact with DNA molecule, DNA sequence. So a core task or core problem of studying transcription regulation using computational approaches is the study of sequence motif. So I'll discuss DNA sequence motif and several mathematical approaches to represent sequence motif, primarily PWM, precision weighted matrix, and several ways of doing motif finding. So from a DNA sequence data to look for sequence motif. And I will mention two types of sequence motif finding approaches: deterministic approach using regular expression enumeration, and the probabilistic approach. It's an EM, expectation maximization algorithm. So transcription factors is one important, very important type of protein molecules that are presented inside the cell nucleus. So it interacts with DNA. As you know, you may or may not know that in mammalian cell nucleus, the DNA molecule is packaged into the structure of chromatin by interacting with a lot of different types of proteins. So some type of proteins called histones are the building blocks of chromatin. So the DNA sequence are wrapped around this histone core to form a small structure called nucleosome. And then, so the DNA sequence can have a few rounds of packing, and then after a few additional rounds of complex high dimensional packing, they can form the structure of chromosome. And this entire chromosome, the structure is very dynamic. So part of the chromosome, they may be open, they may be loose, so that the DNA sequence is not tightly wrapped around the histone, but they expose a part of open chromatin or open part of the DNA, so that other molecules inside the cell nucleus, they're isolated there, they can go to bind or to interact or to bind with this part of the DNA. And this binding process is not random. It has important function to determine how the genome plays this role. So the most important type of DNA interacting protein is transcription factor. Why we call this transcription factor? Because they play a role in regulating transcription. So the transcription is the process of DNA transcribed to RNA. And not all of the genes are transcribed in all the cells, right? Because of the different types of cells, because of different identity of the cells, we only need a subset of genes that need to be transcribed. And transcription factors play a role of controlling which of the genes they need to open and transcribe and when they are going to be transcribed. So I use a diagram here in which this red circle represents the transcription factor so that they can interact with the DNA and at a part of the exposed DNA region. And then they can regulate their target genes either close by or can be very far away from these binding sites. And there are over 1,000 different types of transcription factors in human, for example, at least. So this is several years ago a review paper. I think by the time we believe that the sequence-specific transcription factors are at least 1,600, if not more. I think recent years people keep characterizing more transcription factors. So maybe in the range of 1,000 to 2,000 different type of transcription factors. So many of the transcription factors they also exhibit the tissue-specific or cell type-specific expression pattern. Just like we only need a subset of genes to be transcribed, to be expressed in a particular cell type. Transcription factors are only a certain subset of them are only needed in a given cell type. So in this plot, which is showed is the expression level of the transcription factor gene itself. We can see that many of them have a very cell type specific feature, which means that they are only expressed in one or several selected cell types. So transcription factors are highly cell type specific. And in terms of function, transcription factors have different functions. And usually in the transcription factor protein, they have different structural or functional domains. First and foremost, the most important functional domain in the transcription factor is the DNA binding domain. So all of the DNA specific transcription factors, they will all need to have DNA binding domains. So because they have different type of specific protein domain structure, they can physically interact with a specific structure in the DNA. So that created the so-called transcription factor binding sites. And of course, they have their function, they will also have the effector domain. So transcription factors may have different types of functions. They can regulate transcription factor activity. They can mediate other protein-protein interactions. They can interact with other cofactors or cofactors or other proteins. And they can also have enzymatic activities to give the nearby histone modifications or chromatin structures, adding some chemical modifications to change the potential structure and function of the local chromatin structure with the goal of adjusting the structure of the chromatin and regulate gene transcription or gene expression at a certain stage. And the entire process of how transcription factors interact with DNA and act to regulate, either activate or repress certain gene expression. This process is called transcriptional regulation. And as I mentioned before, transcription factor bind at their binding sites, but their target genes can be far away from its binding sites. If we look at the entire genome DNA structure as a linear structure, so if the transcription factor binding site is very close by, it's next to the transcription start site of the gene, we usually call this area as gene promoters. But if many transcription factors they bind at locations very far away from transcription start sites, we usually call this distal enhancers. And the distal enhancer transcription factor binding, they may be looped into the region close to the transcription start site to activate the transcription process, with the involvement of many other cofactors and other protein to adjust their structure. So to summarize, transcription factors, in terms of their structure, they will have an effector domain to play their function, to play their role, and DNA binding domain, so that they can directly bind or interact with DNA-specific DNA sequences. So the studies of transcription factors will involve these different topics. So we wanted to understand, we wanted to know the expression pattern of a transcription factor. In which cell type does this is this transcription factor expressed, and whether the expression of the transcription factor is cell type specific, and what the next one of the most important question is what specific type of DNA sequence or DNA sequence structure that this transcription factor can interact or the TF can bind. And once we know, or with or without knowing the specific DNA interaction, the DNA structure that they interact, across the entire genome, we wanted to know where across the genome does this transcription factor bind. So genome-wide binding sites is also an important task. We wanted to know for a transcription factor in the specific given cell type. And once we know their binding sites, another important information is their target genes. So which genes does this transcription factor regulate? And other questions includes whether the transcription factor are interacting with other cofactors and whether they will have similar or distinct roles when interacting with different cofactors in different contexts. So these are different important topics when we study transcription factors. So as one way to understand why transcription factors are important in the process of the entire molecular biology, we all know the central dogma of molecular biology is to tell you that the information flow is carried from genetic information in DNA and goes to RNA through a process called transcription, and to the protein, the structure of the protein, through a process called translation. But transcription regulation is especially important because this is a process that to form a information feedback. So different transcription factor protein, they will have regulatory roles in controlling the function of DNA and controlling the function of when and how this genetic information should play a role in different biological contexts. So transcription factors play a role of completing the feedback of information in the central dogma. Okay. Next part, let's talk about motif. So what is a motif? I can give you an interesting example. So this is a fishing shack in the harbor town called Rockport in Massachusetts. There's a small fishing shack called there. Usually this small house is called motif number 1. Why it is called motif number 1 is because in the modern art history, especially in the late 19th century to early 20th century, it is called the most often painted building in America. So we can see the picture in the middle is the picture I took when I visited there. And there are tons of different types of paintings. They all are using this as an object to paint. So most often painted building is called a motif, and called motif number 1. So a motif means a recurring pattern, a distinctive pattern. They carry some feature, but they have to occur repeatedly. So if there's only one, we don't call this as a motif. So generally, that's the concept of a motif. And the second, what is a sequence motif, or in our context, biomolecular sequence motif? So following the same concept of motif, so a sequence motif is a pattern common to a set of DNA or RNA or protein sequence, any type of sequence that share a common biological property, such as functioning as the binding site of a particular protein or a transcription factor. So the recurring pattern of a DNA structure that commonly can interact or can be bind bound by the same transcription factor is called the DNA sequence motif for that particular transcription factor. So why is sequence motif important in the study of transcription factors? We wanted to know what type of DNA sequence a transcription factor can bind, it can go to. So that is the we have the goal of motif finding. So we wanted to identify the potential motif for every specific type of transcription factors. So a common task is that, so we have a type of input data, for example, a set of DNA sequences, and from certain type of experiments. So for example, ChIP-seq. So we didn't talk about the ChIP-seq technology, but just for you to know, ChIP-seq is a genome-wide sequencing-based technology to identify the genome-wide transcription factor, genome-wide binding site of a given transcription factors. So once we know the binding site is, we can grab the DNA sequences around those transcription factors, binding sites, and to form the set of DNA sequence. Or if we know the expression of or differential expression of certain genes, if we perturb the motif, if we perturb the transcription factor activity, then we wanted to understand if we assume that the transcription factor is perturbed, so their binding sites will change their binding sites and hence change the expression of their target genes. So if we measure the genome-wide expression and identify which genes are differentially expressed, we can infer that these potential target genes upstream DNA sequence may contain the binding sites of that transcription factor. So we can also take the upstream sequences of a pattern of gene expression, so that of a gene of a expressed or differentially expressed gene cluster, and from those DNA sequences to identify motifs. Say, for example, usually we may have at least 20 or up to nearly 1,000 different sequences, and each sequences can be from 100 to 5,000 base pairs long. And so that's a typical computational motif finding problem. And this is the input data. And the output data is that we wanted to find motifs or the enriched repeated sequence pattern that occurs in these sequences. So the ultimate goals for biological studies of transcription factor includes from this motif finding, from these sequences. If we identify potential sequence motif, after we identify the sequence motif, we can address the questions including which transcription factors are involved in this process. Because from the existing knowledge or existing database, given the motif, we can predict, we can infer what are the transcription factors, or at least what are the protein DNA binding domains of the transcription factors that the transcription factors have to so that they can bind here. And then what are their binding motifs and what are their effects, whether these transcription factors are activating or repressing the gene expression. And if in certain contexts, we wanted to understand what are the target genes or which genes are regulated by this motif. And also, if there's more in the disease context, if the transcription factor goes wrong, what the disease phenotype can be involved in this transcription factor regulated related context. And also whether there are any binding partners or competitors or cofactors for a motif. So how to represent a motif in a quantitative way? The most straightforward way of representing a motif, a recurring sequence pattern of a motif, is just a regular expression. If we know the consensus sequence, that's the clearest way of representing a motif. Say CA, CAAAA. So that's a very deterministic way or we can make a binary decision whether if it has this sequence pattern, we call this is a motif occurred in this region. But in most cases, transfer factor sequence motifs are not that deterministic. It's not like if it is this exact sequence, CACAA, the transfer factor can bind. If there's one tiny differences on the sequence, the DNA can no longer bind. It's not black and white. Instead, it's more like a continuous pattern. So the chance or the probability of a transcript factor can bind to a certain sequence pattern is continuous. It's not a black and white pattern. So then from a pure consensus sequence, we can allow for some differences, some small differences, small mutations. So people come up with this table of how to about how to represent the sequence. So with some level of degeneracy. So ATGC is the deterministic one. But if there are people use different other letters. So if it's A or G, they use R to represent. For all type of combinations, we give them different letters to represent the degeneracy. And this is the common nomenclature provided by the International Union for Pure and Applied Chemistry. So we have a common way of representing this type of motif sequences. However, there is still one issue. So eventually, using this binary regular expression, we can represent any type of sequence motif, but it is not quantitative enough. For example, if it's an R, we know it's A or G, but we don't know whether it's more likely to be A or more likely to be a G. So we do not have a quantification on top of that. And that's why we wanted a more quantitative approach to represent a motif. In other words, people introduced the concept of position weight matrix, or early years we also called this position specific scoring matrix, PWM or PSSM. So the way of representing a motif using this more quantitative way, using the PWM is like this. So if we want to represent a motif that are, for example, 9 nucleotides long, and here is an example. These are all the possible consensus sequences, all the possible sequences that the transcript factor can interact with. So there's actually an early years, there's a type of specific biotechnology to measure. It's called the protein binding microarray, or PBM. So in that technology, people design different type of short DNA sequences or sequence oligos, and they design a microarray and then added the transcript factor proteins to this entire microarray to see which of the sequences the proteins can bind and give us different signals. So from that type of technology, we can measure what type of DNA sequence that protein, the transcript factor can bind. And once we got the results, so these are all these sequence patterns that this transcript factor can bind, how to generate a position weighted matrix. So first step is to simply count in each position, we count the occurrence or the frequency of each of the 4 nucleotides. So for example, in the first position, so there are 3 A's, right? 1, 2, 3, 3 A's, 2 C's, 1 G, and 4 T's. So we simply count the frequency of every nucleotide in every position. And say, for example, in the fourth position, all of them are G. So all of them are G. So all can, likely, they're all G. So then generate this count matrix. And then we simply normalize this against 1. So then this number tells, so each number in this position, what is the likelihood that this position is for each of the 4 nucleotides. So in the position 1, there are 30% likelihood that this is A, and for the 4th position, 100% likelihood. So it has to be a G, and the 5th position, it has to be a T. So this matrix is called the position weight matrix. Then with the position weight matrix, it is already a very quantitative way of representing a transcription factor sequence motif. And then there's another more visually easier to visualize this motif representation or a graphic representation of sequence motif is so-called the sequence logo. So this we will see this type of sequence logo patterns in many places to represent the sequence motif. So what do they mean? We can see that for a sequence logo, in each position, there are 1 or more letters from ATGC with different height of the letter itself. And also the total heights of each position, they're also different, right? So how to create such a sequence logo? So it's done like this. So starting from a position weighted matrix, how to create or how to generate a sequence logo. So first of all, the sequence logo consists of this stack of symbols or nucleotides. So one stack represents one position in the sequence. And then the overall height of the entire stack in this position indicates the sequence conservation or the conservation level at the position. Or in other words, it's called the information content of that position. And the relative height of each of the 4 nucleotides, each of the symbols within the stack indicates the relative frequency of the nucleotide or nucleic acid in that position. So the entire information content is measured by this few formula. So the information content is the same concept of in the computer science, we use bits, binary bits to give us information. So in the binary case, right, 1 bit means that 0 or 1. So we have 1 piece of information to determine that's either 1 or 0. And a DNA sequence with four possible nucleotides, ATGC, the entire information content is 2 bits. We have this 2 to the 2. So the entire information content is 2 bits. If there is no way to determine whether this is any one of the 4 nucleotides, that means that there's equal probability of all of the 4 nucleotides ATGC. That means that we do not have any information to distinguish any of them. Then we do not have any information. That information content is 0. And this can be calculated precisely using this formula. So for example, if each of the 4 nucleotide, we do not have any information. So the probability of each nucleotide is 1/4, right? So following this equation, log 2 of 1/4 is -2. And then times 1/4 is -1/2, right? The -1/2. And then there are a sum over all 4 possibilities. So then the entire part here is 2. Entire HI is 2. And then 2 minus 2 gives us 0. So we do not have any information content if all of the 4 nucleotides have the probability of 1/4. And we can try another example. If we are sure that there has to be 1 nucleotide, for example, the 4th position, it has to be a G. So it means that one of the four, the probability is 1, the other are 0. So in this case, the log 2 is 0. So the entire H term is 0. So the information content is 2. So we have 2 bits, the entirely 2 bits of information to distinguish among the 4 nucleotides. We are 100% sure that this is only 1 from the 4 possible nucleotides. So any questions so far about how to create a position weight matrix with information content? Okay, very good. Yeah.
So the probability is based on.
Yes, the probability is just based on the position weighted matrix. So, yes, yeah. So FBI is F for B is the base. So which of the 4 nucleotides, 4 bases, and I is the position. I is the position from 1 to N, from 1 to 9. Yeah. So there are actually some underlying physics concepts underlying this entire representation of motifs. It's just the entropy, information content, and entropy. In fact, the H is this formula is the information content. This H equation is the entropy. So entropy is a concept from statistical physics. So that's when physicists developed a mathematical representation to measure very complicated statistical mechanics or thermodynamic structures. And originally, the concept of entropy is proposed by Boltzmann. So this is Boltzmann's gravestone in Geneva. In Vienna. I visited there a few years ago and took this picture. And they have this entropy formula, this equation put on this gravestone. So the Boltzmann entropy is the Boltzmann constant times the log of all the number of microstates. So we can consider that as if we do not have any way of telling what are the possibility of each different microstate, and then that's just the total number of potential states in the big system. And then if we do know the probability, essentially that would be the each state 1 over the number of total states. So that means that it can also be represented by a negative sign after we're taking the log. So that's also the Boltzmann entropy. Another way of representing the Boltzmann entropy is negative Boltzmann constant and sum over the log of the probability times its probability. So it's, in other words, it's the expectation value of the probability distribution. So entropy is actually a very kind of a confusing and at the very beginning a kind of a scary concept, even for physicists or mathematicians. And in the preface of a statistical physics textbook, there's one interesting saying. Ludwig Boltzmann, who spent much of his life studying statistical mechanics, died in 1906 by his own hand. Paul Ehrenfest, who is Boltzmann's student, carrying on the work, died similarly in 1933, and now it's our turn to study statistical mechanics. So it's a kind of a funny and scary opening for studying statistical mechanics and all of the statistical physics concept. But another interesting point is that the concept of entropy is later on introduced into biology, and by a famous physicist, Erwin Schrödinger. And Schrödinger wrote a book called What is Life? So I think it's in 1940s. He wrote this book trying to use modern physics concept, especially statistical physics concept, to explain biology or explain life. And this book actually inspired Francis Crick, who later on discovered the information, the information carrier of DNA and DNA structure. So in the main concept in the entire book of What is Life, proposed by Schrödinger, is that the orderness is an important thing for all living systems. And the orderness, the orderness can be quantified or can be measured by negative entropy. So entropy itself is a way of measuring how it is equilibrium. So there's no information in there, but order is the opposite concept. So all of the living systems, we have very high level of orderness. So if I directly quote what Schrödinger says, so all the living system feeds upon negative entropy. Thus, the device by which an organism maintains itself stationary at a fairly high level of orderliness or a fairly low level of entropy really consists of continually sucking orderliness from its environment. So if we know the second law of thermodynamics is that if in the equilibrium in the isolated equilibrium system, the entropy can only increase, and eventually everything becomes equal, becomes completely average and without any information in it. But the existence of life is the reverse process. So we give them energy, we give them information so that we can create something that have the order. So that's negative entropy. So that's the concept of whole life. And also including everything we're interested in, we are studying in computational biology is to study the order, the orderliness, the information carried in the DNA sequence everywhere in all of the data that measured from living system or biological system. So later on, information scientist, or actually the founder of information theory, Claude Shannon, he transferred this physics concept of entropy into information theory, gives the concept of Shannon entropy or information content or information entropy, which is the same idea of calculating the expectation of information concept. So that which is exactly the same as what we used here. It is the Shannon entropy, and we can convert that into the expression of information content. So actually, this type of sequence logo that with the information content, the base information carries the most information that a position weighted matrix can carry. So imagine that if every position are purely deterministic, so meaning that in every position we only have one possible nucleotide, and then the information are all 2 bits. So then there will be a purely a equal height, a consensus sequence. But then if we have other possibilities in one position, we have multiple possibilities of nucleotides, the entire information is lower, meaning that we have less information to distinguish them, to tell them. And if we, in case that we do not have any information, then the height can represent this. So it is a very good visual way of representing a position weight matrix. Any questions so far? Good. And then, so now that we have a quantitative representation of a sequence motif using position weight matrix, now once we have a DNA sequence, we wanted to know how well this particular sequence matches the position weighted matrix, matches the motif. So there's one way to quantify how well the actual sequence matches a motif or matches a position weight matrix is to calculate such a matrix called the motif matching score, or which is essentially a likelihood ratio score. So the way to calculate this is that this score is the log 2 of the ratio between the probability of this sequence occurred from the position weighted matrix over the probability of the sequence that are from the background. So I'll explain this in more detail. So the probability of this sequence from the background, usually the background is, say, for example, the entire human DNA, genomic DNA sequence. What is the chance of observing A, C, G, or T? So usually it's slightly different from 25% because of the different level of CG content in the genomic DNA sequence. So for example, in this, the entire background we want to use is that the A and T contains 28% and the GC is 22%. So that's like the background. Then how to calculate, so to calculate the probability of a given sequence from the background. For example, if we have this original position weight matrix right here, and we want to calculate the motif matching score of a given sequence, GAGGTAAAC, is that in the denominator, I'll say the denominator first, denominator easier, because it's easier, is that we simply calculate the probability of each nucleotide for their probability in the background. So that's P0. So P0 for G is 0.22. P0 for A is 0.28. So that's the denominator. So it's the background. And then for the numerator, it's a little complicated, but still fairly straightforward. So it's the probability of each nucleotide. What's the probability in the position weighted matrix? So for example, the first position, which is a G, the probability of having a G at the first position is 0.1. So we plug in 0.1 here. And the probability of the second position for having an A, is 0.6. So we plug in 0.6 here. So then, so on and so forth. So we're using this approach to calculate the entire probability of the sequence coming from a certain position weight matrix. And the motif matching score is essentially it's a likelihood ratio score. It's divided the two different likelihood. In other words, what's the chance of the sequence come from this exact motif, or divided by what is the chance that randomly we can observe such a sequence. So this is one way. There are more complicated way to do this calculation, or one can consider this as the zero order Markov chain, because we do not consider the higher order, the interaction between dinucleotides or even three nucleotides. Keep in mind that this likelihood ratio score or motif matching score will be used in the assignment, in the homework for today's lecture. And now let's take a look, how should we do motif scan, motif finding, the de novo sequence motif finding. So we still have the same problem setup. The problem formulation is that we have a bunch of DNA sequence, and we have a goal of looking for common sequence patterns enriched in the input data, in the input DNA sequence. And usually we want to make the comparison with the background, for example, the genomic DNA sequence. And there are two different approaches. So first is the deterministic approach, or regular expression enumeration. So and the other approach is a probabilistic approach using PWM updates, using expectation maximization algorithm, or EM algorithm, or there are using more advanced statistical modeling approaches like deep sampling, which we will not cover in today's lecture. So, for example, first is regular expression enumeration. So the idea is simple because it is a deterministic approach, right? We wanted to check the representation of every here is W-mer or K-mer. So we have a number of K nucleotides. We just want to count how many for each type of K-mer. What are the occurrence of this K-mer in the input data? And the expected W occurrence in the data, we can consider the input data size as well as the genomic sequences, and the observed occurrence in the data, how many of the DNA this K-mer occurred in the sequence, and then the overrepresented W. So which type of K-mer, which type of W-mer is overrepresented in the input data, then compared to the background, will be considered as the binding potential sequence motif in this input data sets. So one algorithm that used this new regular expression enumeration approach to do motif findings called WEDER. That is a fairly old algorithm developed many years ago for DNA sequence motif search. So there are several limitations of this approach, regular expression enumeration. Both pros and cons. First is exhaustive. So it's guaranteed, it's a good thing. We can guarantee to find the global optimum and can find multiple motifs. So if we give a cutoff of how we call this overrepresented K-mer, all of these such K-mer can be considered as a enriched sequence motif. So these can be exhaustive, but guaranteed to identify all of them. But there are certain limitations. They are not as flexible with base substitutions. Like we mentioned before, this just directly use a consensus sequence representation or regular expression approach of representing different motifs. So it's not flexible for if we change slight differences on certain base pairs. And it can generate a very long list of very similar good motifs. And we have a certain limit in the length of the motif we can find, because the length of the motif, essentially the searching space will go exponentially if you increase the length, right? Because all the possible chance of K-mer would be 4 to the K. It's exponential increase. So that's why the more commonly used approach for motif finding is a probabilistic approach. And then I will give you this example of using EM algorithm, expectation maximization algorithm, to do probability as a represented probabilistic approach to do motif finding. So now we formulate the problem and we set the object. First, some notations. So we have a sequence. So we have a sequence data in which we wanted to search for motif. And we have theta 0 representing the non-motif probability or the genomic background parameters. Or in other words, it's just the probability of observing each of the AEGs background as a constant. And the theta is the motif probability matrix parameters, or in other words, we can consider as a dynamically changing position weighting matrix eventually to represent the motif. And pi represents the motif site location. So the problem, if we define the problem as a statistical way, is that given the background, genomic background, theta 0, and giving the input sequence, DNA sequence, we wanted to define or wanted to identify the probability of having certain motif at their location in the DNA sequence. And so this is the entire approach is one type of maximum likelihood approach to estimate it with the goal of estimating the theta that best represents the entire data structure. So the approach of EM approach is to alternately estimate a certain motif PWM, a position weighting matrix, and we want to identify the position of this motif occurrence. And then, given these motif occurrences, we can then calculate what are the most possible motif representation, which is the position weight matrix. And then, and this is the same the object. And EM algorithms and Gibbs sampling approach, they differ in the estimation methods, but the goal is the same. So, for example, so this is a simplified example of an EM approach, expectation maximization approach. So in the E step, in other words, we're calculating an expectation. So expectation is that given a motif, we wanted to calculate the likelihood. So once we have a starting, or we can have any type of starting motif position weight matrix, and then for every position, we can calculate the motif, the likelihood ratio, which is the motif matching score of the actual sequence, how well it matches the position weighting matrix. And for every location, we can calculate a likelihood ratio following the same approach that I mentioned in the motif matching score calculation. And then that's the expectation step, the E step. And then the M step is to calculate a new or to update a new position weighted matrix, is to use the sequence position in every position combined with the likelihood ratio calculated in each position to create a new position weight matrix. So how to do that? So for example, in the first step, if we have a earlier step position weight matrix, and we calculate for the actual sequences, we calculate the first position as likelihood ratio, and the second position have a likelihood ratio, and then we have the likelihood ratios scores and the sequence information of every position, and then so for every position, we can calculate the updated frequency of each of the 4 nucleotides by this equation. So the first position of having a T is to calculate it weighted average. So T at first here is 0.8, and then there's another T is 0.2. So all of the T occurrences times the likelihood ratio, and the denominator is all the likelihood ratios for all the locations, or all the likelihood ratios added together. So in this way, we can calculate AGTC's frequency, updated frequency at each position, and eventually at this step we calculate generate a new updated theta. And then we can do this iteratively until the position weighted matrix does not improve or does not change. And then we obtained both the location of the motif as well as the position weight matrix of this motif. And the related algorithm of this method is a meme. Meme is by far is still one of the most commonly used de novo motif scan approach in computational biology. Okay, so in summary, I gave you a very brief overview of the concept about transcriptional regulation, and I briefly discussed what is a motif and different way of motif representations, a more deterministic way or more quantitative way, and especially using a position weight matrix, PWM, to quantify or to represent motifs. And I briefly mentioned the concept of entropy and information entropy as an important concept in including computational biology and modern biochemistry and biophysics. And for motif finding, I gave you one example of using EM algorithm to calculate motifs. So, okay, that's all I wanted to discuss for today. Any questions? Yes.
So how did you calculate non-motif for non-motif for the
Yeah. Let me okay. Here. Yeah, so the non-motif part is we can consider that as a given constant. It's if we use the genomic background, then the theta 0 for ATGC is constant, right? It's similar like as what we showed here. So theta 0 means that the P0 for A, T, and GC is always constant. So you can consider that the likelihood ratio is to compare the occurrence, how likely this sequence is coming from the position weight matrix compared to how likely this sequence is coming from just a random background. Yeah, the random background is just the curves, the frequencies of each of the 4 DNA nucleotides. Make sense? Yeah. Okay. Any questions? All right. Sounds good. Thank you, everyone.